cardiovascular process. Oh, if you're going, yeah, fire away.
Is the membrane of the thylakoid continuous with the membrane of the chloroplast?
No, it's entirely separate. It's here, and the membrane, the outer membranes of the chloroplast, which are the envelope, they're not connected at all to the thylakoid. I guess if they were connected to the thylakoid, the proteins here, like the cytochrome and the photosystems, would eventually just diffuse into the outer membrane, and then I'm not sure if the process would work if they were in the outer membrane. All right, so all that was light-dependent reaction here, but the whole point of it was to make those ATPs and NADPHs to power the enzymes in the Calvin cycle, and those enzymes need energy because they're doing this non-spontaneous reaction of carbon dioxide into glucose. So let's switch over to that. This is a figure from your textbook that gives an overview of the Calvin cycle. Here they show the Calvin cycle as just sort of one big curving arrow, but that's not accurate. Remember, the arrows often represent enzymes, and I'm not sure exactly how many separate enzymes are in the Calvin cycle, but at least 6 different enzymes I can think of off the top of my head. When you're talking about the molecules that are within a metabolic pathway, we call those the intermediates, and so these things with the little black dots are all intermediates in the Calvin cycle. And what the black dots are actually representing are the carbon atoms of these molecules. So this one right here, that's called RUBP, has 5 carbon atoms, and this one has 6 carbon atoms, and here's an organic molecule with 3 carbon atoms. They're not showing you the other atoms. Those are not supposed to be just chains of nothing but carbon atoms. They have hydrogens and oxygens attached to them also. Different functional groups like OH groups and things like that, and some of them have carboxylic acid functional groups. But just to simplify things, we're just going to focus in on how many carbons each of these intermediate molecules has. That's probably the best way to understand the Calvin cycle. So let's go through these molecules real quickly. One of the molecules we'll be working with here in the Calvin cycle is called RUBP, and you don't have to know what it stands for, but if you're curious, it's ribulose biphosphate. Ribulose is a type of sugar, and it has 2 phosphates stuck on it, so it's ribulose biphosphate. And it has 5 carbons. It's a 5-carbon molecule. And they sometimes use an abbreviation. They say it is a C5 molecule, which just means exactly that it's an organic molecule that has 5 carbon atoms in it. We'll also be working with carbon dioxides, which I guess you could say are a C1 molecule because they only have a single carbon atom to them. There's a 3-carbon intermediate called PGA, which is phosphoglyceric acid. You don't have to know what it stands for, but if you're curious, it's phosphoglyceric acid. And there's another 3-carbon molecule called PGAL, phosphoglyceraldehyde, a C3 molecule. And here they're showing glucose, which is, of course, a C6 molecule. Okay, so those are sort of the characters that we're going to be seeing in this drama of the Calvin cycle. I'm going to show it like this. The green arrows are enzymes, and I'm simplifying. There are more enzymes than I'm showing you here, but these are some of the major ones. And remember, all these Calvin cycle enzymes and intermediates are floating around out here in the stroma. And I don't know if anybody's ever actually counted how many of each enzyme there are, and how many RUBPs, and how many PGALs, and how many PGA molecules are floating around out here in the stroma and are part of the Calvin cycle. If I had to imagine, there's probably thousands, if not millions, of each of these molecules we've been talking about. But just to keep the discussion simple, I'm just going to show you 3 RUBP molecules as part of the Calvin cycle. But what I'm showing you happening to what I'm going to show you happening to these 3 molecules would be the same as if I was showing you the actual thousands or millions of RUBPs that are actually part of the Calvin cycle. Okay, so to begin with, let's start off with imagining that there are 3 RUBPs that are part of this Calvin cycle. Now, in a little while, we're going to have to account for all the carbons that we're going to see going through this cycle. And so don't be afraid to be obvious. How many total carbons are we starting off with here? 15, right? Each RUBP has 5, and if we're going to be looking at 3 of those RUBPs, our total is 15 carbons for starting off with. Now, for a cycle to keep going, so it doesn't run out of materials, eventually we have to regenerate those 15 carbons in this step, and that's why you see some enzymes eventually flowing back in there. And so why I'm bringing this up is, here in a few minutes I want to come back to the fact that we eventually have to feed 15 carbons into here to regenerate those 3 RUBPs that we started off with. Okay, so here's what happens. The first enzyme of the Calvin cycle, this one right here, has a binding site for 1 RUBP. It grabs 1 RUBP, and it also grabs a carbon dioxide. And remember that carbon dioxides float around in the air. They enter the leaf by those little holes called stoma, and they can the carbon dioxides can diffuse into the mesophyll cells and into the chloroplast organelles. So there's a pretty good supply of carbon dioxides floating around. All right, so what this first enzyme does is it carries out a chemical reaction where it joins this carbon dioxide carbon to the RUBP 5 carbons. And so you get this. Now, let me run that backwards. For a brief moment, there was a 6 carbon molecule right there, but that 6 carbon molecule is extremely unstable and breaks apart almost instantly into 2 molecules of this phosphoglyceric acid, this PGA. So essentially what you get is the carbon dioxide is fixed onto 1 RUBP, but instead of getting a 6 carbon or a C6 molecule, it almost instantly breaks apart into 2 C3 molecules, these 2 PGA molecules. All right. Next enzyme is going to take those PGA molecules and, by changing some functional groups on them, change them into PGAL molecules. Oops. There you go. Here's a PGA molecule, a phosphoglyceric acid. You don't have to memorize the structure, just kind of showing it to you. Notice that it has, though, a carboxylic acid functional group as one of its functional groups, which is why it's phosphoglyceric acid. All right, so this enzyme is going to take the PGA, it can take a PGA molecule and change it into a PGAL molecule. This is the enzyme that uses most of the ATP energy and NADPH energy from the light-dependent reaction. Go back. There's a PGAL molecule. Notice it's almost exactly the same, but we no longer have a carboxylic acid functional group over here. So the reaction that enzyme did to change the PGA to a PGAL looks like this. Spontaneous or non-spontaneous? Non-spontaneous, because this was an oxidation or a reduction. A reduction, right. Here's a low energy carbon-oxygen bond. It gets changed into a higher energy carbon-hydrogen bond. I got 1 more minute. So change into a higher energy carbon. That's a non-spontaneous endergonic reaction, uphill reaction. And so that's why you need the ATPs and the NADPHs to power it. It's not going to happen on its own. Let's put the PGAL down there. And now the second PGA does the same thing. It feeds into that enzyme. ATP and NADPH energy is used to carry out that non-spontaneous conversion to PGA, from PGA to PGAL. And now we have 2 of those PGALs down there. And what's going to happen is the other RUBPs are going to do the same thing. RUBP to 2 PGAs. Each one of those becomes 2 PGALs. And I guess it's a good stopping point for now. All right, so we'll pick this up again Wednesday. I will see you in a couple hours.